All right, welcome to lab 2. We're going to go over blood and the heart anatomy. After today, you should be able to name the major components of blood, identify and name the type of cells found in blood, know the functions of different leukocytes, perform and understand blood typing, and lastly, know how and when to use certain blood tests. So blood can be separated into its component parts by using a centrifuge. When a blood sample is taken, it is put into a centrifuge and spun around extremely fast, and this causes the blood to separate into plasma and form elements. Plasma makes up about 50% of our blood volume, and the majority of that is water. Water is extremely important because it is what allows the blood to carry substances like nutrients, hormones, and waste products. Also found in the plasma are electrocytes, respiratory gases, and the plasma proteins albumin, fibrinogen, and globulins. Albumin is responsible for maintaining osmotic balance. Fibrinogen is necessary for the formation of blood clots to stop bleeding, and globulins assist in fighting off infections and the transport of lipids. The other 45% of blood volume is our formed elements. The formed elements are mainly erythrocytes or red blood cells and are what we use to transport oxygen and carbon dioxide. The next major portion of the formed elements are 5 types of leukocytes or white blood cells. White blood cells are the cells that make up the bulk of our immune system. And then lastly, in the formed elements, we have platelets, which are important for blood clotting. This is a chart of all the cell types and the formed elements of our blood. From this chart, you need to know the cell types, their function, and how abundant each cell type is. Don't worry about the specific number, but know the order of which type is most abundant. So our erythrocytes or red blood cells' main function is to carry oxygen and carbon dioxide. We have 5 different leukocytes that are split into 2 categories. We have granulocytes and agranulocytes. Granulocytes contain granules within the cytoplasm, and agranulocytes do not. The first of our granulocytes is the neutrophil. Neutrophils' main function is to phagocytize or eat pathogens and debris. Next in the granulocyte category is the eosinophil. Eosinophils attack parasitic worms and are one of the other factors that play a role in our allergies and asthma. The last granulocyte is the basophil. Basophils release histamine, a signaling compound that is released in response to injury, allergies, and inflammatory reactions. It causes smooth muscles to contract and capillaries to dilate. Basophils also contain heparin, which is an anticoagulant that decreases the body's ability to form blood clots. The first agranulocyte is the lymphocyte. This is what you typically think of when we talk about our immune cell. They directly attack cells and produce antibodies in response to an infection. Monocytes are also agranulocytes and will develop into macrophages that will eat pathogens and debris. And then lastly, we have platelets that only function to plug tears in blood vessels by forming blood clots. So this is a mnemonic that follows the order of leukocyte abundance. The asterisk above each word means that it's a granulocyte. So never is for neutrophil, let is for lymphocytes, monkey is for monocytes, eat is for eosinophils, and the banana is for basophils, which are the least abundant type of leukocyte. This figure shows all the cell types that can be found in blood. So we have red blood cells, platelets, neutrophils, lymphocytes, eosinophils, and monocytes. Red blood cells have about a lifespan of about 100 to 120 days, and they are destroyed in the spleen. One thing to note is that anemia, people with anemias may appear pale due to low hemoglobin, which is a protein in red blood cells that contains iron and is what allows red blood cells to carry oxygen. So this is a close-up image of a neutrophil. Neutrophils have a multi-lobed nucleus, the dark purple areas, and red and blue granules within the cytoplasm. These make up about 50 to 70% of our white blood cells in the body and are the first cells to show up to a site of infection. Neutrophils' main function is to eat foreign invaders. Here is an eosinophil. They have a bilobed nucleus and have red granules. They make up 2 to 4% of our white blood cells and play a role in our allergy response and defense against parasites. Basophils make up less than 1% of white blood cells and also have a bilobed nucleus, but they have dark purple, almost black granules. Basophils release histamine in response to injuries, allergies, and inflammatory reactions, and heparin, which is an anticoagulant. Here we have a lymphocyte. It has a large spherical nucleus and has a thin rim of cytoplasm. And notice that there are no granules within the cell. Lymphocytes make up about 20 to 40% of our white blood cells, and they come in two varieties. We have killer T cells and memory B cells. Killer T cells directly attack and kill pathogens and infections. Memory B cells produce antibodies in response to an infection so that the body can quickly respond if the body is ever infected by the same antigen or pathogen. This is the main mechanism that allows vaccines to work. And lastly, we have our monocyte. Monocytes have a horseshoe-shaped nucleus and make up about 3 to 8% of our white blood cells. Monocytes will develop in the macrophages that will eat infectious pathogens. So now we'll talk about blood test. The blood test check for red and white blood cell count. And there are certain conditions that we can look for. So leukocytosis is an elevated count of leukocytes, indicating an overactive inflammatory response. Typically, we see this when someone has an acute infection. Leukopenia is an abnormally low white blood cell count, which leaves them very vulnerable to infection. And this is typically caused by an acute viral infection, chemotherapy, HIV, or steroid use. Polycythemia is an elevated red blood cell count caused by an increased number of red blood cells or a decrease in plasma volume. This is common in people that live in high altitudes because there is less oxygen and the body must adapt to meet its oxygen demands. And then lastly, we have anemia, which is a reduced oxygen carrying capacity. The first test we'll talk about is the hematocrit test. The hematocrit is a test used to count the number of red blood cells. The book states that hematocrit is used to test for anemia. However, a normal hematocrit may not rule out anemia, but it will rule out polycythemia. A patient may still have a normal hematocrit count, but still show signs of anemia due to low hemoglobin and red blood cells. Normal hematocrit values for males is around 47%, and for females is around 42%. To truly rule out if someone has anemia, a hemoglobin test must be done using a hemoglobinulometer. The average hemoglobin values for males is around 13 to 18 grams per 100 milliliter of blood and 12 to 16 grams per 100 milliliter of blood for females. And this is an image of that hemoglobinometer. This slide shows the mechanism used to stop bleeding. You do not need to know this for lab, but I'll still go over it briefly. When a blood vessel is cut, platelets will begin to stick together, forming a plug. The body will then go through a signaling pathway and release fibrin, which will form a sticky web that will trap red blood cells to form a full blood clot. This test measures how long it takes for blood to clot, and it requires a non-heparinized or a plain capillary tube. Once blood is put into this tube, it is timed and left undisturbed to see how long it takes to form a clot. Clotting is the result of coagulation when strands of fibrinogen combine to form fibrin, which are insoluble and block bleeding. And the last type of blood test is our AB-RH blood typing. This determines the interactions of antigens and antibodies that are found in the blood. This is the test you commonly see done when someone's giving blood or you need a blood transfusion during surgery. So we have 4 different blood types, each with their own specific antigens and antibodies. If you have blood type A, you have A antigens surrounding your blood cells and anti-B antibodies in your blood serum. This means that a person with type A blood will reject type B red blood cells. If you have type B blood, you have B antigen surrounding your blood cells and anti-A antibodies in blood serum, meaning people with type B blood will reject type A blood. If you have type AB blood, you have both A and B antigens surrounding the red blood cells and no antibodies. This means that someone with type AB blood can receive any type of blood and are known as the universal recipient. Type O blood does not have any antigens surrounding the red blood cells, but has both anti-A and anti-B antibodies. Type O is referred to as the universal donor, as any blood type can receive type O blood. However, a person with type O blood can only receive type O blood. And then this chart summarizes everything I said in the previous slide. It also shows the demographic breakdown of each blood type in the US. And then this image shows what happens with each blood type is added to either anti-B or anti-A serum. If coagulation occurs, that means the blood is being rejected and an immune response known as agglutination is happening. And then this is a blood compatibility chart and summarizes what blood can and can't mix. RH factor determines whether your blood is positive or negative. Most people do have RH factor present, resulting in a positive result. Typically, the body does not make Rh antibodies except when Rh negative blood is exposed to Rh positive blood. You usually see this happening in blood transfusions or during pregnancy. So one main issue that arises due to Rh factor is a condition called erythroblast fetalis. This happens when an Rh negative mother has an Rh positive fetus. Rh factor is hereditary, and so this means that the father was Rh positive. Blood mixing between the mother and the fetus will cause a minor immune response, and the mother will start making Rh antibodies. This is usually not an issue during the first pregnancy. However, if the mother has a second pregnancy with an Rh positive baby, these mother antibodies will begin to attack and destroy the baby's red blood cells, which can cause anemia or even death. To prevent this, RhoGAM injections can suppress the Rh factor so that during the second pregnancy, the Rh antibodies do not attack the baby. All right, so now we're starting looking at the heart. We will go over the major anatomical area of the heart, explain the atrioventricular and semilunar valves work, and then trace the path of blood flow. So this is the overall anatomy of the heart. The heart is separated into 4 different areas that will fill up and pump blood, and those are the left and right atrium and the left and right ventricles. There are also several veins and arteries that flow to and from the heart, And the easiest way to go over all of them is to just describe the flow of blood through each structure. So the superior and inferior vena cava are the very large veins that carry deoxygenated blood from the body to the heart, and they both will feed into the right atrium. From the right atrium, blood will be pumped down into the right ventricle. Blood is pumped from the right ventricle up through the pulmonary trunk into the left and right pulmonary arteries, and eventually to the lungs for gas exchange. The blood will return from the lungs back to the heart by way of the left and right pulmonary veins and into the left atrium. On the left atrium, blood is pumped down into the left ventricle. The left ventricle is a very large and strong muscle that will pump the blood into the ascending aorta and out through the brachiocephalic trunk, the left common carotid artery, and the left subclavian artery. And this is how the heart pumps blood to the rest of the body. The heart also has its own set of blood vessels because it is a living tissue and needs its own blood supply. The smaller blood vessels, like the anterior cardiac vein, the straight cardiac vein, and the left and right coronary arteries, will help facilitate cardiac tissue blood flow. Now here we have a cross section of the heart where we can see the inside of the heart, the four chambers, and the different valves we found in the heart. So the atrium and ventricles are also referred to as chambers because they will fill up with blood before ejection. Each chamber is lined with muscle that will contract and push blood into the next chamber or blood vessel. Once the right atrium fills up with blood, both gravity and muscle contractions will push the blood down through the tricuspid valve into the right ventricle. The chordae tendineae act like gatekeepers of the tricuspid valve. When the right atrium contracts, the papillary muscles will also contract and pull on those chordae tendineae and will cause the valve to open so that blood can flow through it. Once the right ventricle is full of blood and ready to contract, the chordae tendineae will relax, closing the tricuspid valve and prevent blood flow from being pumped back into the right atrium, forcing blood up through the pulmonary valve. The same thing happens in the left atrium and left ventricle. Blood returning to the heart from the lungs will pull into the left atrium via the pulmonary veins. Once full, the left atrium will push the blood down into the left ventricle through the mitral or bicuspid valve. Papillary muscles in the left ventricle will pull on the chordae tendineae, causing the bicuspid valve to open. Once the left ventricle is full of blood and ready to contract, the chordae tendineae will relax, closing that mitral valve and prevent blood flow from being pumped back into the left atrium, forcing blood through the aortic valve and into the aorta. Also in this image, we can see the interventricular septum that separates the left and right ventricles. And then we also see the different layers of the heart. So the outermost layer is the epicardium, which is a layer of connective tissue covering the outside of the heart. The middle layer is the myocardium that is entirely made up of cardiac muscle tissue. And then lastly, we have the innermost layer, which is the endocardium, which is a smooth, thin membrane, which lines the inside of the chambers of the heart. And then this is the dorsal side of the heart, and we can see a better view of all the large blood vessels that stem from the heart, as well as the vessels that supply the blood with the heart with blood. Here is a transverse dissection of the heart, where we can see the different valves of the heart. On the tricuspid valve, you can see that we have three different sections or cuspids. That gives it the name tricuspid, as compared to the bicuspid valve, which only has two sections. And remember, the tricuspid valve is found on the anatomical right side of the heart, and the bicuspid or mitral valve is found on the anatomical left side of the heart. And here is a realistic image of the tricuspid valve and the chordae tendineae attached to it. And here you can see the pulmonary and systemic circuits, which are two pathways for blood circulation. The blue is going to represent deoxygenated blood, and red is oxygen-rich blood. So once again, deoxygenated blood returns from body tissues to the heart via the inferior and superior vena cava into the right atrium. From the right atrium, it then goes to the right ventricle to be pumped out of the pulmonary arteries to the lungs. From the lungs, newly oxygenated blood returns to the heart through the pulmonary veins to the left atrium, and then to the left ventricle, where we pumped out to the rest of the body through the aorta. So this is, again, that flow of the cardiopulmonary circulation that I just went over. So again, the oxygenated blood from the body tissue returns to the heart via the inferior and superior vena cava into the right atrium. It then passes through the tricuspid valve into the right ventricle to be pumped out the pulmonary arteries to the lungs. From the lungs, the oxygenated blood returns to the heart through the pulmonary veins to the left atrium, through the bicuspid valve to the left ventricle, where it is pumped out to the rest of the body through the aorta. And here we have the coronary arteries and the cardiac veins. The coronary arteries are going to bring oxygen and nutrient-rich blood to the cardiac muscle tissue. The cardiac veins will remove deoxygenated blood and metabolic waste away from the cardiac tissue. And this is cardiac muscle, the same thing we looked at in Anatomy 1. Remember, the key feature here is the intercalated disc. These intercalated discs join cardiac muscle together and allow them to communicate and allow for synchronized muscle contraction. And this is a cadaver of a sheep heart. The sheep heart is just very similar to the human heart. And the main thing to focus on here, and then you can see the different valves and how they look. And here is another cadaver of a sheep heart showing the inside of the chambers and the structures within them. And here is just another image of the sheep heart. All right, so that's it for lab 2. We went over blood and the heart and circulation. If you have any questions, feel free to shoot me an email. I look forward to seeing you in lab.